Hello guys, welcome back. So let's start with the new short notes video number 8. I hope everything is clear from the previous video. We are always open to doubt, so feel free to comment your doubt, or you can also DM us on the Instagram page we have. So starting with the new organism, that is Streptococcus. First let's learn some basic or general properties of it. Then we'll learn the 3 types of Streptococcus in detail. So firstly, the general properties of Streptococcus is that this bacteria is gram-positive, and in the name itself you can see coccus. Now coccus is spherical, its meaning is spherical. So what will be the shape of the bacteria? It will be coccus shape, or we can also call it as spherical shape. Lastly, a very important point, that these bacteria are capsulated. Okay, so remember these general properties. Now let's divide this Streptococcus into 3 different groups. So it is one kind of Streptococcus is group A, beta Hemolyticus. Okay, the second is a very common one, Streptococcus Pneumoniae. Okay, and last is Streptococcus pyogenes. Now let's learn about each in a bit detail, starting first with the group A beta Hemolyticus. Now what does this cause? This causes angina. What is angina? Angina is pain in the chest. Okay. And what does this contain? It contains O antigen. You should remember, what does it contain? It contains O antigen and M protein. Now what this M protein means? M protein, it is responsible for showing molecular mimicry. Let's know some information about molecular mimicry. Okay? Whenever there is an organism which shows molecular mimicry, means that organism will be initiating an autoimmune response in that person. Okay, so first, what it is molecular mimicry? It occurs between the microbe and the host cell. Okay, so this molecular mimicry will be occurring between the microbe and the host cell, and they share linear amino acid sequence. Means organism which is having this M protein or any protein which shows the molecular mimicry, means that those organisms will have the same linear amino acid sequence as the host is having. Okay, therefore what will happen? Therefore the microbe will start acting or will start mimicking the host cell, right? So when our immune system recognizes that this is a foreign antigen, but then our immune system will get confused because our cell will also be showing the same exact function or will be having the same linear molecular sequence. Similarly the bacteria, so the immune system will get confused. Whom should I kill? Who is the actual antigen? So then the immune system will then kill both, will try to kill the antigen and will try to kill its own tissue. That's why initiating an autoimmune response. Okay, therefore microbe mimics host cell, and this leads to autoimmune reaction. I hope this makes sense. Okay, lastly you should know that to check progress of infection, to check the progress of this group A beta Hemolyticus, we check anti-streptolysin O in the patient's body. Okay, so we look out for this anti-streptolysin O to check for the progression of the infection. Lastly, the treatment, it is a bacteria, so we will be using antibiotics. Specifically if I talk about, then the treatment includes antibiotics which goes by the name benzyl penicillin sodium salt. Okay. Second type of Streptococcus is Streptococcus pneumoniae. Let's know about this. It causes Krok pneumonia. Okay. So this causes Krok pneumoniae. It is a diplococci bacteria. In the question sometimes they mention the organism as Diplococcus. There was a Diplococcus bacilli. So at that time, and it was capsulated. So Diplococcus bacilli plus capsulated, then they are talking about Streptococcus pneumoniae. Okay. The treatment for this bacteria is also penicillin, antibiotic penicillin. Okay. Lastly, Streptococcus pyogenes. What does this cause? This is famous for causing scarlet fever. Now, symptoms of scarlet fever is what you should remember, because it is very, very important, and it is going to come in other subjects also of your Krok. So symptoms include strawberry or crimson colored tongue. So tongue will become very, very red, similar to how a strawberry has its color. So strawberry or crimson colored tongue. And the next property of this Streptococcus pyogen is that it produces Erythrogenic toxin that causes erythematous rash. So the patient will be suffering from a rash which is erythematous. Okay, so it produces erythrogenic toxin that causes erythematous rash. Okay, and we can see this erythematous rash on this patient. Now, how to confirm the diagnosis? So what do we do when we give them antibody against this toxin? Okay, if we give them antibody, so immune reaction takes place and this rash will disappear if the patient is having this infection. If not, then the rash will still be present. So when serum or antibody is injected against this erythrogenic toxin, then rash disappears. Now, if rash disappears, means there was an immune reaction which took place between the antibody and this toxin. So that means the patient is suffering from the Streptococcus pyogenes. So what does this indicate? This disappearance of the rash, it indicates that the patient has this disease. Which one? Scarlet fever. Understood? Now lastly, the treatment, very important. Again, it is penicillin. For Streptococcus group, the best antibiotic is penicillin. I hope this Streptococcus is all clear. Please remember, all three kinds are very, very important and are asked in a lot of different way. So remember all these points which I have said you. The next organism is Trichomonas Vaginalis. Now, if you have watched the medical biology playlist, then you would already know about this organism, Trichomonas vaginalis. We already did there. So for this, you should, in the name itself of the organism, you can see vaginalis. That means it is going to affect the vagina. So how exactly? First, let me write down this, that it causes vaginal problems. So how exactly? Let's know. In this, if we do the vaginal examination, we see strawberry pink vagina, and it is very painful. Strawberry pink vagina, and the patient will complain of immense pain, means immense or intense pain, a very very strong pain in the vagina. Okay, lastly, let's learn something about typhoid fever. So if a patient is there and is suffering from typhoid fever, so if we are suspicious that this patient can be suffering from typhoid fever, so there is a test we do. And then for final confirmation or final identification, there is a different test we do. So let's know about that. So in typhoid fever, write down this point. If we are suspicious, so suspicion, in case of suspicion, we are gonna do diagnosticum test. Okay, the test is known as diagnosticum. It is not diagnostic test, it is diagnosticum test. Okay, please remember this. Now, to be exact, which diagnosticum test? It is passive or indirect heme agglutination. Okay, so in general we call it as diagnosticum test, but to be specific, then it is called as passive or indirect heme agglutination. Now for confirmation or for identification, for final identification test, there is a different test for it. So for final identification test, we do sero identification, and to be exact or to be specific, we basically carry out agglutination. Okay, and in this, what do we see? If it's agglutination test, we will see antibodies. Okay, so remember this, that in case of suspicion of typhoid fever, we will suggest the diagnosticum test. Specifically, if I'm talking about, then it is passive indirect heme agglutination. For if we are carrying out for final identification of a confirmation, then it is sero identification test, specifically agglutination. And what do we see there? We see antibodies. Now these are all short notes of microbiology. I hope this helps and everything is clear. Let's meet in another video. Thank you.